Alrighty, so I'm from a lab in the research side of the hospital where we study the adaptive immune system. And the adaptive immune system is important to us and probably many of you because it's the part of the immune system that physically will change itself depending on the bug that you're infected with. And not only does it change to mediate the best type of clearance to that bug, it also forms memory. So that's the entire idea behind vaccines, right? We get a practice disease that's either killed or just a little part of it, and then we're supposed to be protected in the long run, right? And so the part of the adaptive immune system that we study are the T cells. And the T cells are this really interesting subset of cells that are this lock and key mechanism specific. So even if you have a bug, if it's not what that T cell is designed to target, it won't activate and it won't do anything. And this sort of lock and key specificity is not something that these cells are just gifted with when they appear in our body. It's something that they're educated on. And that education occurs in the thymus, hence T cells. And so during this education in the thymus, they're being told, what's good to respond to, what's bad to target. And it's during this education that they pick up this molecule called CD5. And CD5 was thought to be sort of this, non-functional, just random protein that these cells would express. But here, recent studies have started to show that based off of the amount of CD5 that they express, there's actually consequences when it comes to the infection. And so what you can see on the top is that CD5 high T cells tend to be a little bit stronger at recognizing their pathogen. And when this turns into an infection, what it means is that they're a little bit better at clearing the initial burst of the infection. And so what my lab is really interested in is trying to define why do these cells hold on to that CD5 expression? Why is it so important after they exit the thymus? And then after we understand that, what is the consequence during an infection? What are these cells actually doing with this CD5? And then finally, I don't really have to be a medical doctor to tell you that there are big differences between me and a baby, right? So in babies, we know that their T cells express comically high levels of CD5, and you can see that on the bottom with just the massive amount of little sticks on it. And we know what we want to do is once we find out all of those answers in the adult system, we want to go back into babies, and we want to try to see, can we manipulate their T cells so that maybe they can form memory? And they won't get as many repeat infections, and they won't have such a hard time with things like influenza and RSV that we as adults don't really think about. And that is the basis behind my work. Thank you.